This lecture is on gel electrophoresis. This was one of the main things covered on the air practice test, and I thought it would be a great thing to go over before we take the air test. So we're going to dive into this. This is part of Unit 3, which is our DNA unit and heredity. This is figuring out whether we have DNA in our system, what kind of DNA we have present. So we're going to go through what gel electrophoresis is and also talk about what its uses are. So if we're moving through here, what is gel electrophoresis? This is a technique that is commonly used in laboratories, some universities, different places like that, where we're going to separate the charged molecules like DNA and RNA and proteins according to their size. The charged particles are going to move through the gel when we have an electric current that passes through it. The gel is positively charged on one end and then negatively charged on the other. That's going to allow the movement of our particles across the gel. So this is a picture here of what gel electrophoresis system looks like. If you can see, my pointer here has the electrode. There's a negative electrode and a positive electrode on the other side. Here are the slots where we place our DNA. Typically, you're going to have a slot of a known solution. Then you're going to have slots here that contain your DNA sample that you're testing. And you're going to turn on the electrode, and the DNA is going to move according to whether or not it is negatively charged or not. DNA is negatively charged, so it'll move towards the positively charged electrode. As you can see over here, we are separating out our A, C, G, and T. So if you can remember that, adenine, thymine, cytosine, and guanine, based on their bands and electric charge. That will tell you what kind of DNA that you have. So what is this used for? So our applications, we can do DNA fingerprinting. We can also detect genetic variants. So what we mean by that is if we have different strands of DNA or whether organisms with a DNA strand, they can detect different variances amongst those organisms. You can also use detections of pathogens. If you don't know what a pathogen is, it is something that infects your body, whether that be a virus or bacteria of some sort. And right there it says bacteria strains. So they can tell the difference between different bacteria strains. Maybe one bacteria strain is harmful for your body. The other one may not have an effect. So gel electrophoresis can tell you what type of strain that you have. So the example I'm going to talk through here using gel electrophoresis, this was a project I did in my microbiology college class. This was analyzing. What we had to do was go and take samples at different portions of our university, and what we were trying to see is if we had Staphylococcus DNA present in our samples. Most of you would think, listening to the word Staphylococcus, that's what staph is. So like a staph infection would have Staphylococcus DNA in it. So what we did was we took two samples of a swab area. Our swab areas are labeled M1 and M2. That was taken from one portion of our university. And then P1 and P2 are another sample taken from a different portion of the university. So we took those swabs. We did a PCR, which we will not go into in this section here. But we did that to break apart the DNA from our bacteria from our sample. And then we place our samples in these units here, or those slots on our gel electrophoresis slab. So we have M1, M2, P1, and P2. We're going to compare it then to what's known as the marker. The marker is a known Staphylococcus DNA sample, and we're going to compare everything to that marker. So if something shows up in our samples that is comparable to our marker, we would be able to say that there is some form of Staphylococcus DNA in our samples. So looking at our results, we turned on our gel electrophoresis and we let the DNA separate going towards the positive electrode. As you can see, the marker has 1, 2, 3, 4, 5, 6, 7 strands separating in that DNA. If you look at our P1 and P2, we do not have any strands of DNA being present. That tells us that Staphylococcus is not present in our P1 set of samples. If we look at the M1 and M2 slots, however, though, if you go down M1 here, M1 has one band similar to that of the marker. M2 does not have any strains. But we can look at M1 and say because a line showed up in our gel electrophoresis, we can say that there is a commonality of the DNA found in M1 compared to our marker. So we can say that Staphylococcus could be present in our M1 sample there. So that's how we can use gel electrophoresis when we are looking at our different samples here. Okay, so in class then we will go over different examples of air testing questions that go along with gel electrophoresis, and hopefully this helps us when we are preparing for our air tests.